Good morning, friends. Good morning, students. Today we will discuss metabolism of methionine, cysteine, and cysteine. Right. So first of all, we start with methionine. This video will cover introduction to all of these three. Then we will discuss transmethylation reactions. Then we will discuss synthesis of cysteine and cysteine from methionine, and then we will discuss degradation of methionine. Right. And then finally we will discuss disorders of this metabolism. Right. Now, first of all, I start with introduction of methionine. It is a glucogenic amino acid, right? Glucogenic, and it is a essential amino acid, right? Now we'll start with the products which we can synthesize from methionine, right? First, the products of methionine. First is coenzyme A, right? Then it can also be used for the synthesis of taurine, right? Taurine can be further used in bile salt synthesis, right? Then it is used for the synthesis of creatine, right? It is also used in the synthesis of cysteine and cysteine, right? Then it is also used for the synthesis of PS, phosphoadenosine phosphosulfate, that is used for the synthesis of mucopolysaccharides, right? Now we will start with transmethylation. Transfer of methyl group from active methionine to acceptor is known as transmethylation, right? For example, we have methionine. It is activated to form acetinosyl methionine that can donate methyl groups, and this is active form, right? Now we'll discuss all the transmethylation reactions in detail. For example, we have methionine. It is first converted to acetinosyl methionine, that is active methionine, right? So this reaction requires methionine adenosine transferase, right? So it requires ATP, and it is converted to pyrophosphate and phosphate, right? This reaction is irreversible reaction, right? So now we are having acetinosyl methionine, that is active methionine, that can donate methyl group. So when it will donate methyl group, the methyl group will bind to acceptor, right? That is used in methylation reactions. For example, we have norepinephrine. When it will combine with methyl group, it will be converted to epinephrine. One of the examples of reaction. And these reactions require methyl transferase, right? Now we got acetinosyl homocysteine. It is further converted to homocysteine by adenosine homocysteinase, right? We'll get reaction by adding water, and adenosine will come out, right? Now we got homocysteine. It is converted finally back to methionine, and this reaction requires B12, and the enzyme is again methyl transferase, right? And there is conversion of tetrahydrofolate to tetrahydrofolate, right? So this reaction requires B12, and it is PLP dependent. Then finally it is converted to methionine. And when we have deficiency of B12, this reaction will not proceed, and it is also known as folate trap, because methyl tetrahydrofolate will accumulate and it is not converted to tetrahydrofolate. And when the reaction proceeds, we will finally get methionine. So these are four major intermediates: methionine to acetinosyl methionine, then acidinosine homocysteine, then homocysteine, and would finally get methionine back. Now we'll start with synthesis of cysteine and cysteine from methionine, right? So these are the four basic reactions: methionine to SAM, SAM to acidinosine homocysteine, and then homocysteine, and finally conversion back to methionine. So we have homocysteine here. It will combine with serine, right? Right? With the enzyme beta synthase, right? It will be converted to then cystathionine, right? This reaction is B12 dependent again, right? Cystathionine will form cysteine by cystathionase reaction by the enzyme, right? Cysteine is a monomer, and cysteine can be further converted to cysteine. The second, our third amino acid, 2-cysteine will form disulfide linkage with each other to finally form cysteine, right? Our third amino acid, right? Now cysteine will further combine with homoserine to form propionyl CoA, right? Propionyl CoA will form succinyl CoA. Succinyl CoA now can enter TCA cycle, right? So earlier we discussed it is glucogenic amino acid. Now it is leading to the synthesis of glucose because the products are leading to the synthesis of glucose via TCA cycle. Now the third part is degradation of cysteine. So we have cysteine here. First of all, it can undergo decarboxylation reaction, right? Decarboxylation reaction. So carbon dioxide is removed. Will get mercapto ethanol mine, right? This will lead to the synthesis of coenzyme A, one of the products of this metabolism. And cysteine can also lead to the synthesis of two products, that is cystic acid and sulfide, by reaction with cysteine dioxygenase, right? So we'll get two products, that is cystic acid and sulfide, right? Cystic acid can lead to the synthesis of taurine, right? Taurine is used for bile salt synthesis, right? Then sulfite is used for synthesis of phosphoadenosine phosphosulfate, and it is used for mucopolysaccharide synthesis, right? And cysteine can also lead to the synthesis of pyruvate, right? Pyruvate can enter TCA cycle. So this amino acid is glucogenic, as we discussed earlier. So there are basic four products. One is coenzyme A, second is taurine, third is PAPS, phosphoadenosine phosphosulfate, and fourth is pyruvate, leading to the synthesis of glucose. Now the final part, the disorders of this metabolism. There are three major disorders. First is cystinuria, second is cystinosis, and third is finally homocystinuria. It is further classified into four types: 1, 2, 3, and 4. Right. So first of all, we start with cystinuria. Actually, it is due to the defective absorption of these four amino acids: COAL. C for cysteine, O for ornithine, A for arginine, and L for lysine, right? So due to defect of this absorption, the disorder is cystinuria, and there is accumulation of cystine stones in this patient. And the second disorder is cystinosis, that is cystine storage disease, and there is accumulation of cystine in reticuloendothelial system like spleen, bone marrow, and kidneys, right? So there is accumulation of cystine in the reticuloendothelial system, organs like spleen, bone marrow, and kidneys. And finally, coming on to the third disorder, that is homocystinuria. It is due, the type 1 is due to deficiency of enzyme homocystathionine bit synthetase, right? So we have high levels of homocysteine in these patients, right? So high levels of homocysteine can lead to myocardial infarction, stroke, and pulmonary embolism as well, right? The patient might suffer from osteoporosis and mental retardation as well, right? And homocysteine interferes with collagen synthesis, so there will be more production of LDL, and LDL will lead to these conditions like MI and stroke. And the other categories are type 2, that is due to deficiency of enzyme methyl tetrahydrofolate. Type 3 is due to homocysteine methyl transferase, and type 4 is due to defective absorption of vitamin B12. If B12 supplementation is given, it can solve the problem. So that's cover the complete metabolism of methionine, cysteine, and cystine. And keep studying. Please like the channel, subscribe the channel, and do share with your other friends as well. Thanks a lot.